Hello dear student, as I promised in absorption part 1 I already discussed passive diffusion system of transcellular absorption system. So today I will start the second part, that is active transport, that is also comes under transcellular. Then I will go for intracellular or intercellular, then I will go for vesicular transport. That second two topics are very small because I already have told you that 90% of drugs got absorbed by their transcellular absorption method and only 10% goes through or absorbed by different to inter and vesicular transport. So they are not that much important, so we can cross it in easily or in small time. So the first thing I will discuss, that is active transport. That I already have discussed what is active transport. Active transport is also a transcellular absorption method where that drug is got absorbed through the one GI epithelial cell. What is the difference between passive and active? I already discussed. In active, the drug is generally moves opposite or against the concentration gradient. So here is requirement of energy, and we know in body for energy the main source is ATP. So there can be direct use of ATP or no direct use of ATP according to how that ATP is required, direct or indirect. Through that, active transport is also divided into two parts. One is primary, where that direct ATP is required. Direct ATP is required. The second part is secondary. Here ATP is required, but indirect ATP is required. No direct ATP is required. That secondary also is divided into two parts. One is symport. Second one is antiport. So first I will discuss about primary. I have already told you in primary active transport system the energy is given by direct ATP, and we have to remember in primary active transport system generally ion or molecule absorbed into our body who are distributed or who will be ionized after taking into our body. So after taking into our body, that ionized or ion molecule drug generally go against the concentration gradient, and their form and their absorption way is one directional. That means drug can be absorbed against the concentration gradient in one direction, they can't revert back. So they are one-sided or unimolecular or one path or unipath. That's why that is also called uniport. So that active transport system also coded uniport because here the flow or absorption of molecule or ion is unidirectional or one directional. They can't revert back into the backside. So now I am going to discuss about secondary. Here no direct ATP is required because they generally take advantage of previous concentration gradient. But here the difference between primary and secondary is unidirectional. That means that molecule or ion can be flow in one direction. But in secondary they can be moved towards one direction. That means towards or one-sided, like I am telling two ions are there, one or two. That two ions will flow together in same direction or same towards, and some drugs or molecule can move towards itself or opposite side. According to that secondary transport system is divided into two. One is symport and the second one is antiport. So by picture we can easily understand the symport and antiport. Let's suppose that is a GI epithelial cell. By picture we can easily understand what is symport and what is antiport. Let's suppose two epithelial cells are there, that is 1, that is 2. We are supposing two ions are there, sodium ions and potassium ion. In symport, what will happen, that both ions will travel in same direction. That means Na plus will go travel here, and that K plus will travel K, that, or it may be that upwards, but that will be in same way. So that is called as symport. In antiport, that one drug, let's suppose Na plus will go that side. That means that K plus will go opposite side, or vice versa. If Na plus is going that side, K plus will go down side. That is called an antiport. So by here we are finishing off that passive and active diffusion, which comes under transcellular absorption of drug, and that is the main pathway of drug absorption, because I already have told you 90% of drug got absorbed by that transcellular absorption mechanism, and remaining 10% is going through paracellular or intercellular, and second one is vesicular. By that only 10% drug got absorbed. So the second topic I will go to discuss is paracellular or intercellular. I already have told in paracellular or intracellular, international. That means between two epithelial cells, the drug got absorbed. So in that also two parts are there. Absorption through tight junction of epithelial cell, and the second part is absorption or sorption. What is tight junction? Tight junction is same like pore transport system, where the little pores are present, and that through that pores the drug got absorbed. But in tight junction, they are also present as pores, but they are present pores which are little bit bigger in size than that pore transport, and they are very tight junction. Through that tight junction, drug got absorbed in between two endocellular or endocellular cells. But in part absorption method, that also drug got absorbed in by that little pores, but here that little pores are not permanent pores, they are temporary pores. That means in endocellular cell, sometime in between the two cells, sometime in endocellular membrane, little pores appears for temporary time. Through that temporary pores, that drug got absorbed. So according to that, how that pores is, they are defined into tight junction and paraabsorption. In paraabsorption, pores are temporary. That means they comes for some time. After drug absorption, they get vanished. But for tight junctions, that pores are already present, like pore transport, but here the pore size is little bit bigger than pore transport. So I already have covered transcellular and paracellular. Now the third point is vesicular transport system or vesicular drug absorption. That is not that much important, because only 1 to 2% drug got absorbed by that vesicular absorption. How that vesicular absorption works, that is same like diffusion. Here also drug moves towards concentration gradient. That also use of concentration gradient, and they got absorbed through different pores present in the cell membrane, and through that cell of membrane permeability goes through, and drug got absorbed by that vesicular membrane. Here also energy is required, energy is not required. By that process, generally different type of vitamin got absorbed in our body. That can be water soluble vitamin like A, D, E, K, or water soluble vitamin like vitamin B complex, C, K, everything, that all pass through or absorbed by vesicular absorption system. That vesicular absorption is classified into two parts, that is pinocytosis and phagocytosis. It is differentiated, or it is classified how that membrane is engulfed. Engulfed means how it is eating or drinking that drug, and it depends upon how the drug material is. Is the drug is solid in state, or liquid in state. According to that, they are differentiated in two parts. One is pinocytosis and phagocytosis. First one is phagocytosis. We can easily understand by a small diagram. That is cell membrane. It is cell, that is cell membrane. We are supposing one drug we are taking that is solid in part, solid in state, that is solid drug. Now that membrane will eat it. So it is also called as solid eating method or solid eating absorption process. Here what will be that cell will make a hole or will change its membrane, will change its structure. So what it will take, it will attract that solid part and it will try to engulf it, and after some time that drug will be completely engulfed into that cell and that is solid. So that is called as phagocytosis. And here like cell is engulfing or eating that solid drug particle, so that method is also known as solid drug eating or solid drug eating process. The second one is pinocytosis. In pinocytosis is also the process is same. Here also cell is there, but drug here is that is liquid form. And here that drug is liquid in nature, so it is called liquid drinking. We generally drink liquid and we eat drug or we eat that solid material. Last time that drug was in solid, so that engulf of solid process or phagocytosis is called as cell eating process, and here it is called as cell drinking process because the drug is liquid. Then the remaining thing will be same, that we will engulf the liquid particle inside it and that drug will be stay here, that is the liquid part, that is called as pinocytosis. So we have to remember phagocytosis is called as cell eating because here the drug is solid. In pinocytosis the system is same like phagocytosis. Here the particle is liquid, the drug is liquid, that's why that is called cell drinking. So that is called cell eating, that is phagocytosis because there is solid particulate is there, and here that is called as cell drinking because the here drug is liquid solute. So by that I have already finished up different process that is involved in absorption of drug in two successive classes, and by that I covered that full absorption process by which that drug got absorbed into our body. By that same process we will discuss about distribution, elimination and metabolism in different parts so that you can understand easily or you don't have to mug up all that topic into your body, because that all are important subject and important topic before going into that main topic. So you have to first understand how that absorption metabolism works. So I will go very slowly that processes two classes so you can easily understand, and you can ask me whatever problem you are facing when you can ask me whatever problem you are facing when you are studying that absorption process. That I will clear first in the next topic. Then I will move forward for distribution, elimination and metabolism in two parts. So what I have told you today about absorption, that I have already cleared as a handwritten note, which I will going to upload in that video, and that link will be down into the description of Google Drive. From there you can study absorption and you can ask me whatever question or whatever problem you have into the next classes when I will go forward for the distribution part.